All right, let's get right into it. Today, we're talking about some truly incredible news coming out of the world of cancer research. A team at Guangxi Medical University has published a study with a strategy so clever, it honestly sounds like something straight out of science fiction. So let's break down how they're using a pig gene to set a trap for cancer. So, first things first, we have to ask the biggest question. Why is cancer so tough for our own bodies to fight off? I mean, think about it. Our immune system is this amazing, powerful defense force, right? It's constantly on the lookout, taking out invaders. But cancer? Well, cancer is a whole different ballgame. It's a rebellion from the inside. And this really gets to the core of the problem. You see, foreign invaders like viruses or bacteria, they stick out like a sore thumb. Our immune system spots them and goes on the attack, no problem. But cancer cells are sneaky. They're our own cells that have just gone rogue. So they are absolute masters of disguise, tricking our immune system into thinking, hey, nothing to see here, just a normal cell. This is also why treatments like chemotherapy can cause so much collateral damage. It's hard to target just the bad guys without hurting the good guys too. So that leads to this really brilliant idea. What if you could somehow force those disguised cancer cells to wear a giant flashing neon sign that just screams, I'm the bad guy? You know? What if you could paint a target on them that's so big, so obvious, so foreign, that our immune system would have absolutely no choice but to see it and attack? And this, right here, is the stroke of genius at the heart of this new therapy. The researchers thought, okay, let's do exactly that. They figured out a way to make cancer cells express a gene from a pig. Yeah, you heard that right, pig cells. By making our own rebellious cells look like they come from a pig, they trigger one of the most violent immune reactions our bodies are capable of, hyperacute rejection. It's the very same powerful response that makes organ transplants between different species almost impossible. Okay, to really wrap our heads around how this works, let's use a super simple analogy. Let's think of your immune system as the police force, and cancer as a gang of criminals hiding out somewhere in the city. So first up, you've got the criminals. Those are our cancer cells. They're hiding in plain sight in a regular neighborhood, and they look just like all the other normal citizens. The police, our immune system, are patrolling, but they can't tell who's a criminal and who isn't, so they just drive on by. Now, here's where it gets clever. The scientists send in what we're going to call the mice. Now, this isn't a normal mouse. It's a special virus that is really, really good at getting into the criminal's hideout. Why only the criminals? Well, think of it this way. Normal, law-abiding cells have a home alarm system. If a virus gets in, the alarm goes off and they immediately call the police. But the criminals, the cancer cells, they had to disable their own alarm system so the police wouldn't notice them in the first place. So the mice can sneak in, and the criminals can't call for help without exposing themselves. And this is the final brilliant piece of the trap. The scientists attach a super alarm to these mice, and that's our pig gene. Once the mice get inside the criminal's hideout, this alarm goes off. And it's not a quiet alarm. It's an incredibly loud, powerful signal that the police force simply cannot ignore. It basically screams the exact location of the hideout. So what happens? The police swarm the location, raid the building, and take down every single criminal inside. The virus finds the cancer, and the pig gene calls in the cavalry. So let's connect that back to the real science. What are these mice? Well, they're a type of virus called an oncolytic virus. The specific one they used in this study is a modified version of something called Newcastle disease virus. It's actually a virus found in birds, but it's been engineered so it's really good at targeting cancer cells and leaving our healthy cells alone. And that huge police raid? In scientific terms, that's the hyperacute rejection we were talking about. It's the super intense, immediate immune attack our bodies launch against foreign tissue. What's so amazing here is that the scientists took something that's normally a huge problem, a major bug in organ transplants, and they cleverly turned it into a cancer-killing feature. And this attack is a devastating one-two punch. First, you get the direct assault from the immune system wiping out the cancer cells. But second, the immune reaction is so intense that it also causes tiny blood clots to form in the blood vessels inside the tumor. This essentially cuts off the tumor's supply of food and oxygen, starving it from the inside out. Okay, so the theory is just fantastic, right? It's elegant, it's clever, but the million-dollar question is always the same. Does this thing actually work in the real world, in a living being? Well, let's take a look at the data. The first signs were just stunning. In preclinical trials with monkeys that had liver cancer, the therapy was incredibly effective. As you can see, tumors shrank dramatically. In some cases, they disappeared completely. I mean, gone. This was the huge green light that they were really onto something big here. So, based on those amazing results, they moved on to a phase 1 clinical trial in humans. And it's really important to understand who these patients were. This wasn't for early-stage, easily treatable cancers. This trial was for patients with very advanced, late-stage cancers that had already spread and had basically stopped responding to all other available treatments. So, what was the big headline number from the human trial? An incredible 90% disease control rate. Just let that sink in for a second. 90%. For patients who had pretty much run out of options, this therapy had a positive effect for 18 out of 20 of them. This means their disease either stopped getting worse, it started to shrink, or in one case, it went away completely. That is a truly remarkable result. Let's break that down a bit more. One patient, one person, had what's called a complete remission, meaning doctors couldn't find any visible tumors anymore. 6 more had a partial remission, so their tumors shrank in a big way. And for another 11 patients, their disease became stable, which is a huge win because it means the cancer stopped growing. Only 2 patients didn't respond. These are just incredibly hopeful numbers. Now, as incredibly exciting as all of this is, we have to take a deep breath and be responsible. We have to ground ourselves in the reality of how science works. This is an amazing step, but it's a process, and that process is long and careful for very good reasons. This timeline really helps put it all into perspective. The amazing results we've just talked about, they're from a phase 1 trial. A phase 1 trial is mostly designed to see if a new treatment is safe. We are right here. The next steps are much, much bigger trials. Phase 2 and Phase 3 to prove that it's truly effective for lots of people before it could ever be approved as a standard treatment. So let's just be absolutely crystal clear on a few things. This is incredibly promising early-stage research, but it is not a treatment that is available to the public right now. There's a lot more work to do. And please, please, be very careful of scams online from people claiming they can sell you this therapy. They can't. It's not real. What this is a major, hopeful, and exciting step forward, but it's a step on a much longer journey. It's not the finish line yet. You know what's so cool about this therapy is how it combines a smart virus with an even smarter way to unmask cancer to our own immune system. It really shows how creative and clever science is getting in this fight, and it leaves us with a really exciting question to think about. As we get better and better at understanding the basic rules of biology, what other clever traps are out there just waiting for us to discover?